Why do we yawn? Almost certainly not for the reason you think. Yawning is one of the most common and mysterious human behaviors. We do it when we're tired, bored, waking up, or even just seeing someone else yawn. But despite its ubiquity, science has long struggled to explain why we yawn. And according to a recent article in The Guardian, the answer might surprise you. The myth of oxygen deprivation. For decades, the prevailing theory was that yawning helps us take in more oxygen when our brains are sluggish. The idea was simple: a deep inhalation boosts oxygen levels and wakes us up. But recent research has largely debunked this explanation. Studies show that oxygen levels in the blood remain unchanged before and after yawning, suggesting that something else is at play. A cooling mechanism for the brain. One of the most intriguing modern theories is that yawning helps regulate brain temperature. Neuroscientists have found that yawning may act like a radiator, cooling the brain when it gets too warm. This could explain why we yawn more in certain situations, like when we're stressed, tired, or transitioning between states of alertness. Yawning might be the brain's way of maintaining optimal performance. Just as a computer slows down when it overheats, our cognitive functions may dip when our brains get too warm. A yawn, then, could be a built-in cooling system. The contagious yawn, a social signal? Another fascinating aspect of yawning is its contagious nature. Seeing someone yawn, or even reading about yawning, can trigger the reflex in others. This phenomenon is especially strong among close social groups, like family and friends, and may be linked to empathy. Some researchers believe that contagious yawning evolved as a form of nonverbal communication, helping groups synchronize their behavior. In this view, yawning isn't just a physiological response, it's a social one. So, why do we yawn? The truth is, we still don't know for sure. But the latest science suggests that yawning is far more complex than a simple oxygen boost. It may be a multi-purpose tool, cooling the brain, signaling social connection, and helping us transition between mental states. The real reason we yawn is almost certainly not for the reason you think. It's a reminder that even our most mundane behaviors can hold deep evolutionary secrets. Unusual fossil discovery rewrites the history of freshwater fish. A tiny fossil has made a big splash in evolutionary biology. Recent research reveals that a 67-million-year-old fish fossil is rewriting the story of how freshwater fish evolved, and it all comes down to hearing. A fossil with a hidden message. Scientists from the University of California, Berkeley, and other institutions have uncovered a fossilized fish with a remarkably well-preserved Weberian apparatus, a bony structure that connects the swim bladder to the inner ear. This structure enhances hearing sensitivity and is still present in 2/3 of all freshwater fish species today, SCI Tech Daily. The fossil, dating back to the late Cretaceous period, shows that this advanced hearing system evolved much earlier than previously thought. It suggests that ocean-dwelling fish entered freshwater environments multiple times, each time adapting in unique ways. Hearing across the ages. The Weberian apparatus is a game-changer. It allows fish to detect subtle vibrations and sounds underwater, giving them a sensory edge in murky or noisy freshwater habitats. The discovery of this structure in such an ancient fossil indicates that complex hearing systems were already in place before many modern fish lineages emerged. This challenges the long-held belief that freshwater fish evolved their hearing abilities gradually over millions of years. Instead, it appears that these adaptations happened rapidly and repeatedly as fish migrated from saltwater to freshwater environments. A global journey. The fossil also sheds light on the global migration patterns of ancient fish. As the supercontinent Pangaea broke apart, fish species dispersed across newly formed rivers and lakes. The repeated evolution of the Weberian apparatus in different lineages suggests that freshwater environments exerted strong selective pressure for enhanced hearing. This discovery not only deepens our understanding of fish evolution but also highlights the dynamic relationship between environment and adaptation. Fossil of carnivorous swamp monster found in Brazil. A remarkable fossil discovery in southeastern Brazil is shedding new light on the ancient predators that once roamed South America. Paleontologists have unearthed the lower right canine of a massive carnivorous mammal from the Tremembe Formation, an extinct creature that may have dominated its swampy habitat millions of years ago. Meet the sparassodont, South America's apex predator. The fossil belongs to a probaryenid sparassodont, a member of an extinct group of metatherian mammals that filled the role of top terrestrial predators during the Cenozoic era. These fearsome creatures were endemic to South America and evolved through two major diversification events, first in the Eocene and later in the Miocene. This particular specimen dates back to the Deciduan age of the Oligocene epoch, roughly 29 to 21 million years ago. Measuring 5.73 centimeters (2.3 inches) in length, the canine shows signs of heavy wear, suggesting it was actively used by an adult animal to capture and process prey. A window into the Oligocene. The Tremembe Formation, located in the Taubate Basin of São Paulo state, is the only late Oligocene fossil site in Brazil known to preserve mammal remains. Its rich fauna includes metatherians, bats, rodents, armadillo relatives, and several extinct ungulate groups. The discovery of this sparassodont adds a second record of the Proborhyenidae family in Brazil and expands their known geographic range. It also highlights the Tremembe Formation as a vital paleontological resource for understanding the final stages of the Paleogene period in tropical South America. Swamp monster of the past. Based on the fossil's location and morphology, researchers believe this predator lived in a swampy or lacustrine environment and likely occupied the role of apex predator. Its powerful teeth and adaptive features suggest a life of active hunting and dominance in its ecosystem. Anteavis crurilongus, a surprising predator from the dawn of dinosaurs. A newly described dinosaur fossil from Argentina is reshaping our understanding of early theropod evolution. Named Anteavis crurilongus, this small but agile predator lived approximately 231 million years ago during the Carnian stage of the late Triassic, and it had features far more advanced than expected for its time. A fossil that challenges the timeline. Discovered in the Esquigualasto Formation of northwestern Argentina, the fossil includes cranial and postcranial remains that reveal a streamlined skull, elongated lower legs, and a compact, muscular hip structure. These traits were previously thought to belong only to much later neotheropods, suggesting that dinosaur evolution was more diverse and dynamic from the very beginning. The genus name Anteavis means before bird, while crurilongus refers to its long legs specifically, a femur that's 80% the length of the tibia and a metatarsal 3 at 63% of tibial length. This anatomy points to a fast, ground-level hunter with efficient stride mechanics and lightweight bones built for speed. Climate and dinosaur diversity. The fossil was found in a layer just above a level dated to 231 million years ago, in a region that experienced a shift from humid to semi-arid conditions. Researchers believe this climate change may have driven early dinosaur diversification, followed by a temporary decline in abundance around 228.91 million years ago. Fossil records show a rebound in diversity about 15 million years later during the Norean stage. This pattern suggests that climate played a key role in shaping dinosaur evolution, with Anteavis representing a crucial data point in the early radiation of theropods. Why it matters. The discovery of Anteavis crurilongus expands the known anatomical spectrum of early theropods and challenges the idea that advanced features emerged gradually. Instead, it supports the view that early dinosaur evolution was marked by rapid experimentation and ecological adaptation. Yale scientists solve a century-old brain wave mystery. For over a century, neuroscientists have puzzled over the rhythmic waves of brain activity known as gamma oscillations. These fast, synchronized patterns are linked to perception, attention, and memory and are often disrupted in conditions like schizophrenia, bipolar disorder, and Alzheimer's disease. Now, researchers at Yale University have finally uncovered where gamma waves originate and how they influence behavior. Cracking the gamma code. Led by Dr. Jessica Carden, the Yale team developed a new method called CBASE, clustering band-limited activity by state and spectrotemporal feature, to measure gamma activity with unprecedented precision. Unlike previous techniques, CBASE captures short, discrete bursts of gamma waves rather than assuming a continuous flow. This allowed scientists to map gamma events to specific behavioral moments, such as decision-making or sensory detection. Their findings revealed that gamma waves don't arise solely in the cortex or thalamus, as previously thought. Instead, they emerge from dynamic interactions between the two regions where sensory input from the thalamus is amplified by the cortex. This discovery challenges long-held assumptions and opens new doors for understanding how the brain processes information. Behavior and brain rhythms. To test the role of gamma waves in behavior, researchers trained mice to respond to visual stimuli. When they disrupted thalamic signals, gamma activity in the cortex faltered and the mice performed poorly. Conversely, when gamma patterns were artificially replayed into the brain, mice responded as if they had seen the stimulus, even when they hadn't. This suggests that gamma waves are not just background noise, they actively shape perception and decision-making. Toward early detection of brain disorders. Because gamma activity is altered in several neurological and psychiatric conditions, the Yale team believes it could serve as an early biomarker for diseases like Alzheimer's. They're now investigating how neuromodulators like acetylcholine and norepinephrine, both lost in neurodegeneration, affect gamma patterns. If successful, this research could lead to non-invasive diagnostic tools that detect cognitive decline before symptoms appear. Scientists discover a molecule that links aging throughout the entire body. A groundbreaking study has identified a molecule that may act as a master regulator of aging across multiple organs. This discovery could pave the way for new therapies that target aging at its root, potentially extending health span and delaying age-related diseases. The molecule, a systemic aging signal. Researchers at the University of California, San Francisco, UCSF, have found that a molecule called prostaglandin E2, PGE2, plays a central role in coordinating aging throughout the body. PGE2 is a lipid compound involved in inflammation and cellular signaling. As we age, its levels rise in various tissues, including the brain, liver, and muscles. The team discovered that elevated PGE2 suppresses mitochondrial function, the energy-producing machinery of cells, leading to fatigue, cognitive decline, and other hallmarks of aging. By blocking PGE2 signaling in mice, scientists were able to restore mitochondrial activity and improve physical and mental performance. A unified theory of aging? This research supports the idea that aging is not just a collection of isolated breakdowns in different organs, but a coordinated process driven by shared molecular signals. PGE2 appears to act as a broadcast molecule, sending aging instructions throughout the body. The implications are profound. If we can modulate this signal, we might be able to slow aging systemically rather than treating individual symptoms organ by organ. Toward anti-aging therapies. The UCSF team used a drug that inhibits the EP2 receptor, a key target of PGE2, to test their theory. Mice treated with the drug showed improved muscle strength, better memory, and enhanced endurance. These results suggest that EP2 inhibitors could become a new class of anti-aging medications. While human trials are still a long way off, this discovery marks a major step toward understanding and potentially controlling the aging process.